think what's important is that APDA has always been there for me and for us. I also mentioned that John's mother was diagnosed after John. So we had both a young onset and then a more traditional grandmother, if you will, in our family that were dealing with the different ages and stages of this challenging disorder. When John was first diagnosed for us, we were looking for lots of information. We knew we had a lot to learn. And the APDA was there. And in fact, we were pretty young as an organization here in the Northwest. The organization was founded quite some time ago out of our headquarters in New York. But our chapter here in the Northwest has always been really scrappy. And that's a good thing. We are a grassroots organization with people who really, really care, that are connected to the cause. They either have Parkinson's themselves or a loved one who does. And our board members are the same. I have found in my professional career that there are some boards you can sit on where people just want to make motions and approve and pass things on paper. Our group is really the type of people who get our hands dirty and are really looking for new ways to make APDA relevant to everyone in this room. So for us, it started with information. Information is power. There's been great questions today, many that we're able to address from a research perspective, given the topic of today's sessions. But also, if you haven't attended other APDA functions, I would encourage you to do so. For those who are newly diagnosed, we have a Good Start program. It really talks about what to expect. If you're new to the community, how to get engaged with support groups. We have a number of support groups around the city, around the state. And in fact, I don't know if you know, but the reason we are APDA Northwest is because we serve a 5-state region. We're gonna test my skills here. Idaho, Montana, Washington, Oregon, Alaska. So if any of y'all have friends up in Alaska, make sure they're getting our information. So of course, beyond education, for those who have been living with this disorder for a while, you don't need the Good Start program, but maybe you're interested in, for more advanced, sorry, what's my name, Jen? Sorry, Take Control. Take Control is for those who have been living with the condition for a while longer. We also do an annual Optimism retreat. This is our first research symposium, and I think the rhythm will most likely be that we'll either do the research symposium or the retreat each year. But again, another day where we're together from 10 in the morning till 3 in the afternoon, figuring out how to face these challenges together. One of the things that I've loved from the very beginning is that we focus on hope and optimism. We've felt this for so long, I will tell you, that we put it in the tagline. We literally redid, the branding out of National was redone just a year or two ago, and they took a lot of input from chapters, and we said, you know, our programming is about hope and optimism, and we feel like that's what needs to sit underneath our logo. Everything that we do is about education, events, fundraising, research, and opportunities for you to feel connected to a community that can support one another. A couple quick mentions of those events. We just finished our annual gala. It's a lovely evening. It sells out with about 500 participants. We auction off all kinds of crazy things. There's games and events, and we honor one individual or family who has given a great deal to the Parkinson's community. It's a great event. If you've never been before, maybe mark your calendar for next spring. We raised over $500,000. It was $480,000 for everyone in this room, right? I mean, that's a victory for everybody here. The other big event that's coming up, our second largest fundraiser, is the Optimism Walk. And Allison, who was hiding last time I tried to introduce her, there she is in the back waving. Allison is in charge of our walk. It's taking place on June 2nd. Who already has their team signed up? Oh my goodness. Okay, who doesn't live in Seattle? You got, each of those hands represents a virtual walk from wherever you live. I want you to know if you don't live in Seattle, you can still sign up. You can ask friends at work, at church, through your kids, whatever may be, your neighbors, and actually raise funds and be a virtual participant. So I would encourage everyone to take a look. It's on our website, apdanorthwest.org. So that's taking place on June 2nd. It's an exciting community event. It's super fun. The walk is very gentle path along the beautiful water. And we have quite a party at the end. Suzanne Cameron with Cameron Catering, her husband has been living with Parkinson's for 20 years as well. The delicious food today is from Cameron Catering. She does the food for our galas and also will be doing the food for the walk. She's spectacular. So literally down to what you're eating has a tie to Parkinson's. How about that? That's right. Round of applause. That's right. We also have a program department. We put out grants as a board of directors. We vote on supporting particular programs in the communities across the 5-state region. Those include yoga, boxing, music, dance, tai chi, a whole variety of physical activities, keeping that muscle memory alive and movement. Whether you're standing or seated, there are some fantastic programs for fitness, seated fitness as well, and just general coordination. Who's taking advantage of those amazing programs out there? That's awesome. Next year, I want to see 3 times as many hands. And then, of course, the last piece is research, as we have mentioned many times today. It's such an essential part of what we do, and it is something that sets us apart. We are the only local organization that's funding research. You've heard of many others talking about Michael J. Fox Foundation. There's other great work being done on a national level. The only local support of research is APDA funding the registry. So I do hope that you'll sign up, and if that junk mail comes, just consider it a reminder that we're working for that cure. So back to business. Who's ready to get back to business? I'd like to welcome to the stage our next speakers. They're going to be speaking about a year in review of Parkinson's research, which I'm excited to hear about. I'm going to go back and get my pen to take notes. Dr. Jason Aldred is a movement disorder neurologist from Spokane, and he is the co-owner of Selkirk Neurology and the owner of Inland Northwest Research, which also is a silver level sponsor. It's got a table out in the walkway, so I hope you'll go out to the atrium before you leave us today. And to Dr. Cyrus Zabatian, did I do it right, I hope? And he's here with us from, he's a professor of neurology, and he's here from the University of Washington and the VA of Puget Sound. He's also the co-founder and current director of the Washington State Parkinson's Disease Registry, of which I keep mentioning and is very important. Lots of gold stars on my list here. So please welcome Dr. Aldred and Dr. Zabatian.
Great. Can you hear me in the back? That thumbs up, thumbs down? Better? I think that thumb's going up.
Close. Hold it really close.
Okay. I'm a bit myopic, but I think those thumbs were up. So I'm going to talk today. I'm a neurologist and neurogeneticist. And our task today was to talk about things that were new and exciting in the research world in this past year or so. Yep. I'm about to swallow it. Okay. Can everybody hear me now? All right. So, again, our task today was to talk about some of the most exciting things that have happened in the world of research in Parkinson's disease over this past year or so. And there were many, many things that happened, but I'm going to focus on one specific topic, and that's precision medicine. So what is precision medicine? Precision medicine is the idea that if you look at a patient population, this could be any disease. And the idea is that everybody who has a single disease isn't the same. There are subtypes of diseases. And why does that matter? It matters because, for instance, if you can't distinguish these different subtypes and you give an entire group a treatment, that treatment might be very effective for a small subgroup of people. It might be extremely effective in 20%, but the rest of the 80% of those folks may have very little benefit or side effects, and you've really wasted resources and time by treating people who were unlikely to respond. Well, it sounds great. Why don't we just sort all this out? Well, the idea is that we have to have markers or a way of picking these different subtypes out. So when we do that, and if we're able to do that, what we can then do is that we can group all those people together. So all the people in blue here would respond to treatment A, and they'd respond to treatment A very well, and not to any of the other treatments. And if we can stratify all of these patients into the right groups, and then target a treatment that works just for their group, we've saved time, we've saved effort, we've made patients much happier, and this sort of theme can potentially hold true for any sort of disease. But there are areas where this has been in practice for years and other areas where it's just coming to bear. And the best example of this is cancer. So in many different types of cancer, and I'm just using breast cancer as an example, you take a piece of tissue and you look for receptors that are expressed on that cancer tissue. And so, for example, if your cancer is estrogen receptor positive, you get a different treatment than if it's negative. If you have human epidermal growth factor receptor, HER2, you get a different treatment. And these treatments are targeted for these particular receptors. So these receptors live on the cancer and make it grow. And so you will get, depending on which receptor you have, you'll get targeted treatments that bind to that receptor and shut it down. Okay? And so this idea has been going on for years in cancer treatment. But for diseases like Parkinson's disease and Alzheimer's disease and ALS, it's been a hope that we'd eventually be able to bring this sort of more precise approach to treatment, but it hasn't really happened until now. And so what's happened in the past couple of years is that we've started to be able to understand how we can start sorting out these groups. And how can you sort out groups? Well, there's lots of ways. So genetics is one of them. We can look to see if certain people have mutations in certain genes. There's lots of other ways. So in theory, we may be able to differentiate different types of Parkinson's disease by proteins you have in your blood or your spinal fluid. We might be able to do this by looking at things that you've been exposed to, your imaging, so MRI or PET studies. There's a lot of different ways that we could, in theory, do this. I'm going to simply focus on one area because that's where I think we have the most traction right now, and that's genetics. So many of you have heard of the GBA gene. Actually, I'd like to show of hands. How many people have heard about the GBA gene in Parkinson's disease? Just give me a raise. Just raise your hand if you have. So a few people, not too many people. So the GBA gene encodes this enzyme, glucocerebrosidase. And if you get a copy from your mother, copy from your father. If you get two defective copies, you get this disease, Gaucher disease, which is a disease where an abnormal lipid builds up in cells all over your body. It makes your spleen large, it makes your liver large, it can crowd out your bone marrow, cause bone pain, cause your blood cell counts to go low. If it's a severe form, it can even cause death in utero or as a child. And a small subset of those folks get Parkinson's disease. But what we and others have found over the years is that if you have one defective copy, and people in this group who have one defective copy are much, much, much more numerous than these folks, it increases your risk for Parkinson's disease. And there are over 200 mutations in the gene. Some of them knock down the function of the enzyme a little bit. some of it a lot. So if you have a mutation that knocks it down a lot, you are even more likely to get Parkinson's disease. If you have a defective copy that knocks it down a little, you're at risk, but less so. Important to remember that just because you have one of these mutations doesn't mean you're going to get Parkinson's disease. It just increases your risk. There's probably other things that are going on with your lifestyle and your environment that combined with one of those mutations pushes you over the edge and gives you Parkinson's disease. So this enzyme, to get down to the weeds now, this enzyme, glucocerebrosidase, does one thing, and that's that it takes this compound, glucoceramide, which is a lipid basically that's in the plasma membrane of our cells all over our body. It's something we need, we have, it's part of our bodies. And it breaks it down into, it takes this glucose group off and creates this other molecule, ceramide. So when you have a defect, when this enzyme is defective, what ends up happening is this. It's just like when your sink starts backing up. This glucosylceramide molecule starts building up, and it starts building up, and it becomes a problem for your body. And the idea is that, and the idea is, here's that molecule. As this molecule starts to build up, there's something called a lysosome, which is an organelle in all of your cells that's essentially there for waste removal. It gets rid of proteins that have gone bad, and this is constantly going on all the time in all of our bodies. When this molecule, glucosylceramide, starts building up, it starts gumming up the lysosomes. They aren't working as well, and so other proteins start building up. That's the theory. One of those proteins that starts building up is alpha-synuclein, and you'll hear more about that in a little bit. But the idea is that it's this glucosylceramide building up that's causing a lot of the problems, eventually causes nerve cells in your brain to be sick like dopamine neurons and eventually die. So what can we do about this? So the difference here is that because we've known about Gaucher disease and that it's a genetic disease for decades, there's been many decades of research on it. So we have a head start. And so there have been a number of different things that you can do you can replace the enzyme for people with Gaucher disease, because you're mostly interested in what's happening in other areas of the body. But with Parkinson's disease, you can't just give somebody new enzyme because it doesn't cross into the brain. So you need to come up with other molecules. So here's an example of 3 clinical trials that are going on or have recently finished. So one of them is from Sanofi Genzyme. It's a glucosylceramide synthase inhibitor, and so it maps onto the map here. So if this is glucosylceramide, one of the things you can do to get it down is to actually inhibit the enzyme that makes it in the first place. So if you hit this enzyme, you can decrease the amount of glucosylceramide you have, and in theory, try to get things back in balance. So this has been in a phase 2 clinical trial that's ongoing right now called MOVES PD. I believe the Booth Gardner Center is a site for that study, as well as OHSU. The second compound is called LTI-291. This is an effort between Allergan and LTI Therapeutics. And these drugs essentially activate glucocerebrosidase. So one of them actually binds the good copy of the enzyme and increases its activity. The other acts as a chaperone, and that's this drug here, Ambroxol. I'll talk about that in a minute. But the idea here is that it's revving up the good copy of the enzyme and causing it to over-synthesize and overact. And then this drug, Ambroxol, is a drug that's been around for decades, and it was a drug that's been used for respiratory disease to thin your mucus. And people found surreptitiously that it seems to have an effect on glucocerebrosidase by taking the mutated enzyme and chaperoning it or following it around and making sure that it doesn't get broken down as fast as it otherwise would. and this is a drug that's off patent and is being studied by a number of investigators. So here you have targeted therapies that are really meant to see if the first studies are going to be to see if they work in people who have these mutations. And LRRK2, probably more of you have heard of LRRK2. Let's have a show of hands for LRRK2. Okay, so quite a few more of you have heard about LRRK2. So LRRK2 is a gene that we found not because it was associated with another disease, but because of its association with Parkinson's disease. So there are several mutations in the gene. This is the gene. There are several mutations that have been described. A couple of them actually were first described in families right here in Washington State. And the most common mutation is this G2019S. And essentially, this curve simply is here to show that if you carry one of these if you carry the G2019S mutation, you have somewhere between a 25 and 40% chance of getting the disease by the time you reach 80, which means that the majority of people who have this mutation and live to that age don't get it. And again, why? It's probably because of a combination of other genes and things in your environment that we don't entirely understand yet. So the idea here is that this gene, there are different mutations, but what they all seem to have in common is that they affect this part of the protein, which is a protein kinase. And protein kinases basically go around adding phosphate groups to proteins. And why is that important? Because that's the way that proteins often signal each other to turn on or turn off. So by adding that phosphate group to a bunch of other proteins downstream, you can start causing all kinds of other proteins to do things. And if you overactivate or underactivate some of those proteins, you can cause a number of things to happen. And the idea is that nerve cells are particularly vulnerable to these actions, and so they take a hit. And if they go below a certain level, of course, you get Parkinson's disease. So here, most of the research has been trying to find small molecules that will turn down that kinase activity. So it's still working, but it's not adding phosphate groups as overactively as it once was. And there's one study that was just completed, a phase 1 study. This was just in healthy subjects. The company is Denali Pharmaceuticals, and this DNL201 is a small molecule that does just what I said. And this is a study essentially where phase 1 studies, you hear us talk about phase 1A and 1B and 2 and 3 studies. So these early phase 1 studies essentially are there not to see if the drug actually slows down Parkinson's disease, but what the properties of the drug are like, oftentimes in healthy controls. So these are just some pharmacokinetic data showing how long does it take if we keep giving you the drug for it to reach steady levels in your blood. Does it get into spinal fluid? If so, how much? Does it seem to bind to the molecule, so-called target engagement? Does it bind to the right molecule, and does it seem to cause any changes in that molecule? So that study was completed, and now a phase 1B study is just underway. So we know that this doesn't look like it harms people, and the next question is it going to actually do anything beneficial for the disease? That's an oversimplification, but. And I just want to point out that what's exciting about these drugs is that they are going to the heart of the problem. They are going into the cell at the molecular level, and in theory, hopefully, addressing the root of the problem. And so, and hopefully, if that's the case, they're not just going to treat the symptoms of Parkinson's disease like levodopa or dopamine agonists. They're actually, we hope, going to slow down the disease. This is just a diagram that sort of shows what we think happens, again, an oversimplification, when these are dopamine neurons, and as with normal aging, we all lose dopamine neurons. And if we start losing them too quickly, at some point, we get to the point where we start having early symptoms and then obvious symptoms, and then our disease gets very severe. The idea with neuroprotection is that you slow this process down. So if you catch it early, you may be able to delay the time at which somebody actually gets symptoms. And if they already have symptoms, you might slow how fast their symptoms get worse. And that is the holy grail of all neurodegenerative disorders. We don't want to just treat the symptoms. We want to prevent them or slow it down. And that's what people mean by a quote-unquote cure. So one last point that people often ask. So how many people does this affect? And I have this pie diagram here to simply show you that. So if you were to take a random sample of people across the US who have Parkinson's disease, about 1% of them are going to have a LRRK2 mutation. About 8% of them are going to have a mutation or other variant in GBA that matters. So that's about 9% of people that these studies are going to pertain to in theory. So if they work, they could help about 9% of people with Parkinson's disease. Now, it's possible that these molecules will actually help people who don't have one of these mutations. We don't know yet. The studies are being initially targeted to people who do, but it's possible that they might help people who are part of this, the rest of the pie. And that would be wonderful if it happens. We just don't know yet. And I'll end by going right back to the beginning, which is, here we go with this. As I mentioned before, we want to stratify people based on a subtype of the disease, and we can do that a number of ways. And perhaps we've reached the point in Parkinson's disease where Group A is defined by a GBA mutation. Group B is defined by LRRK2. There's another mutation in another gene that causes early onset Parkinson's disease. Perhaps those people will be a different group that respond to a specific targeted therapy, and on and on. And it isn't, again, that these different groups have to just be defined by genetics. We know that probably in a lot of people, genetics plays a role, but not an enormous role. So there are other markers that we need to and are continuing to look for to be able to stratify these groups and target the treatments to the groups where they would really do the most good. Okay, so with that, we're going to save questions to the end. Sorry. We're going to save questions to the end. Thanks.
Well, can everyone hear me back there? I'm going to try to put the mic uncomfortably close to my mouth up here so you can all hear. Well, my name is Jason Aldred. I wanted to first thank the APDA for inviting me out here and you all for joining us today. I am from Selkirk Neurology and Inland Northwest Research. It's my private practice and medical research company based in Spokane, Washington. It's Eastern Washington. Is anyone here from east of the Cascades? A couple back there and hopefully maybe some more online? Okay, excellent. And also, I teach medical students in the second year of movement disorders and Parkinson's disease at the 4-year WWAMI program based in Spokane, and Washington's newest medical school, Washington State University, based in Spokane. So work with both of those schools talking to those students about Parkinson's and whatnot. Well, today I'm going to talk about therapies that may slow Parkinson's progression, next generation therapies, and we, in theory, have been saying that for years. But Dr. Zibatian's talk already today, I think, has shed light on that we're in a very different time now for Parkinson's research. Specifically, his conversation about precision medicine, specialized medicine of sorts, is certainly one of the exciting new angles for Parkinson's research. And there's still a whole need for everything else, the other group, right? Ways getting at patients that don't necessarily have LRRK2 mutations in GBA. So we're trying to do therapies that might work on many different angles, for different patients. I want to talk about two different kind of categories here. One of these, the first one is the immune system and immunotherapy. And so just as a brief introduction, our immune system is really second only in complexity to the human nervous system in the human body. So there's, learning this in medical school, we learn about all the many pathways of the nervous system and all the many pathways that the immune system can go down. And it, to the point of perfection, in a way, our body uses its immune system to fight off external invaders, virus, bacteria. And also, we don't really recognize this as much in the general public, but our immune system's crucial for controlling cancer and other things within our body that doesn't go properly. And so when we're talking about neurodegenerative disorders and systems within cells in our brain that are supposed to function appropriately, when they do not function appropriately, hopefully there's some kind of gap mechanism to fill that in. And that's where we're going to be focused with immune therapy and trying to actively direct that. So again, immunotherapy has revolutionized our treatment of autoimmune disorders and cancer. We use mechanisms similar to the body's own defenses, so using our own innate tools to specifically target the unwelcome protein, hint, hint, alpha-synuclein, perhaps, or LRRK2 or some other mutant proteins. And then, this is where it's important, right? I mean, to be able to do this is one thing, but to be able to dose it, to really titrate it, to individualize the therapy, to allow for a careful reduction in the amount of abnormal protein to prevent further damage. Because, as you've heard already, many of these proteins they're supposed to be there. They're just not behaving appropriately, or because of a variety of reasons, they are formed in ways that can put us at risk for Parkinson's. Alpha-synuclein. Raise your hand if you've heard of alpha-synuclein. Okay, we're getting even more hands up there progressively. So when we're kind of coming to a unified theory or something that unifies Parkinson's, there's truly nothing truly unified in Parkinson's, but many things approach that. And alpha-synuclein is certainly one of those, an abnormal protein that we have normally in our bodies, but abnormal in Parkinson's. In particular, the way that it's kind of clumped. So I'm using the term clumping to just refer to Parkinson's disease forming kind of at-risk structures here, this alpha-synuclein monomers. Monomers here. The alpha-synuclein monomers are normal functional proteins that perhaps help nerves transport things, and we don't exactly know, but function appropriately. However, there are, for certain circumstances, not completely clear, but certainly gene mutations would be one of them, alpha-synuclein oligomers. So they kind of clump up, and then this can be transported outside of cells and put us at risk for developing these fibrils, and those go on to form Lewy bodies. And who has heard of Lewy bodies? I think that beat alpha-synuclein for hands, so we've made it. So Lewy bodies are certainly associated with decreased cell survival, cell death. But the interesting thing here is before even getting to Lewy bodies, we see that these alpha-synuclein oligomers, so these clumps, right? Clumps don't sound good, and they're not good in our cells, but they place our dopamine cells at increased risk for cell death. Just a little technical piece here. Alpha-synuclein is something called the Brac Hypothesis. So I was a fellow at OHSU when the Brac Hypothesis came out. This is a theory about the spread and progression of Parkinson's. We know that abnormal alpha-synuclein and Lewy bodies are not healthy for us and put us at risk for Parkinson's and are associated with the actual clinical features of Parkinson's. But why do people with Parkinson's progress at different rates? We heard in many of these meetings there's thousands of different types of Parkinson's, one for every kind of person or more. And really there is some truth to that of sorts, but there are factors that influence progression. And so why do some people on one end of the bell curve progress really rapidly, but they still have Parkinson's, and other people progress extremely slowly over decades? And that's due to some factors within our bodies that must vary. And perhaps the progression may relate to the spread of alpha-synuclein oligomers, or these clumps, from one cell to another, killing them. We have premotor symptoms of Parkinson's. So these are symptoms of decreased smell, acting out dreams at night, bowel movement issues, other problems as well, mood symptoms. And these clumping of proteins are found in cells outside of the dopamine system as well. Again, getting to the point of actually changing the progression of Parkinson's is not going to focus solely on the dopaminergic system, the dopamine system. It's going to focus on many different systems. Ultimately, things that really relate to two of the greatest fears of people with Parkinson's is not having mobility issues and serious thinking issues. So those are two of the big ultimate factors of Parkinson's beyond what we can currently treat pretty well that we really have a need to get at. The idea, though, coming back to this alpha-synuclein and Brac hypothesis, is the moving of abnormal protein from one cell to another. This may actually provide a unique opportunity for treatment. This is just a picture of what happens on the top slide here in Parkinson's disease when we see, supposedly within cells, alpha-synuclein here, monomers here, these are okay, then they start to clump and they spread from the affected cell outside of the cell, clump even more, aggregate, clump even bigger into the Lewy body. So using an immune system type model, what we are starting to see interest in and results from many, many years of folks in the lab is these clumps going outside of the cell and then being bound with antibodies. Antibodies, of course, are produced by our own immune cells, but can be synthetically produced as well, targeted directly to something that we want to isolate and harness and tag onto other immune cells in the body to say, hey, take this out, let's remove this before it aggregates and causes toxicity to our dopamine system and the other cells and spreads. So again, a way to slow progression in Parkinson's. Again, the idea here is that there are certain types of glia, there are certain types of immune cells, microglia, that get activated in Parkinson's, and we've heard about immune activation or inflammation. You know, inflammation is seen in Parkinson's disease, and the thought at some time was maybe that was somehow a trigger, or maybe it's an after effect. Well, when dopaminergic cells and other cells in Parkinson's are affected, there is definitely a response when cells die. So it could be that you might target one specific, the spread of a Lewy body from one neuron to another, but if you don't also dampen down the inflammatory pathways, you could still have other damage to other cells. So in Parkinson's mice, we see that overproduction of this protein, alpha-synuclein, there's increased microglia activation as well. And so this the glia are supposed to digest. They're kind of the garbage can, if you will. They're supposed to digest this abnormal protein, but when it becomes clumped big enough, they no longer can do that. They can no longer achieve their goal of removing this out. So if we can get to these protein clumps before they get too big, for lack of a more precise word, this might really allow us the point to intervene from the spread from one cell to the other outside of the cell. Now, when we start to talk about immunotherapy, we think about other things like vaccines and what, and the term Parkinson's vaccine's been thrown around. That was the first way that I remember in looking at a paper from Austria where they used an actual active vaccination, looking at ways to target alpha-synuclein. Well, certainly it would be nice to have a one and done type thing, or maybe a vaccine with a booster shot. There are some problems with that, so I just wanted to speak to this in this presentation. There's some variation between immune systems, and particularly, you know, it just so happens that people with weaker immune system are often older, and you know, some people respond differently to vaccines, so there's a lot of variation in response. And in the small studies that have been done so far, one of the concerns is that, like our immune system varies in how we respond to a vaccine, it may vary considerably in the response to like some type of therapy targeted to make the body's own system recognize the alpha-synuclein and remove those. Really what we're talking about now in terms of using an immune therapy is more in line of like something we call passive vaccination. And again, this is more appropriately termed immune therapy, but I wanted to bring this point out because I find patient after patient will kind of bring to me this concept of this vaccination, and I wanted to put it in those terms. So given after exposure to an agent, like a rabies vaccine in this case, so rabies is a virus that can go from cell to cell and cause terrible damage to the nervous system. So after you're exposed to something like that, if you're given a treatment to isolate that and to limit the spread of the disease, that can kind of be this therapeutic window. In Parkinson's, the therapeutic window we obviously want to get to as early as possible. But our hope with any of these therapies that target abnormal proteins is we can get to them at any point in Parkinson's. The later we get there, the less effective they may be, but nonetheless, at any point, certainly earlier is better, but at any point it could potentially be effective. These types of treatments must be given frequently enough to neutralize the damaging substance. So there is some frequency issue here. So as opposed to like a injection, a vaccine type thing you get where you're one and done, or you get a booster, the things that are passive vaccinations that don't rely on the body's own immune cells to really generate the initial antibody have to be given periodically. So that's kind of a little bit about how they're different. So just very briefly, our clinical trial group in Spokane is remarkably large and efficient for the size of our area. And so we've got a number of clinical trials. I'll talk a little bit more about them later, some of the other ones, but the phase 2 clinical trials currently looking at this type of technique is the SPARK trial. So we were part of Biogen's phase 1 study, and also the phase 2 SPARK trial where we're looking at a compound called BIB54 in people with Parkinson's disease. And we're looking at safety and outcomes. We're looking a bit at disease progression as well. You heard Bob talking just a little bit about that. This is the Biogen trial here. There's another trial by Roche called the Pasadena Trial, and this is looking at a different type of antibody that binds to the different end. So there's different ways to grab something, you know. So the Biogen antibody is grabbing the alpha-synuclein from one end, and the Pasadena trial from Roche is grabbing it from the other end. So this is a really neat idea. Don't just try one thing only one way, but try a certain therapy like immune therapy from different angles to try to see which one is the best effect. In general, these studies are relatively similar in that they're monthly infusions of these monoclonal antibodies with measurements for safety and clinical effect compared to placebo for over 1 year, and then another year where everyone is converted over to the active infusion. So this is just a kind of a summary here. It's just a little bit redundant, but not only are we here in the degenerative state seeing the death of neurons, but also overactivation of these microglia, the immune type cells in the nervous system. And the hope is that with immunotherapies, we can neutralize with the antibodies neutralize alpha-synuclein before it triggers spread neuronal death to the next neuron, and also immune activation. 3:10 today, I know what I'll be doing, hopefully. Go Zags. Tyrosine kinase, this is my last piece here, tyrosine kinase inhibitors in Parkinson's disease. Who has heard of c-Abl? Right, okay, we're back down again. So c-Abl is a common protein in any parts of cells with a wide range of function. This is something until, a couple years ago, I last read about in medical school cancer type courses and whatnot and cell biology courses. It's effective, or it's present all over the body. It's kind of a regulator type protein. Proteins do work in cells. In neurons, it involves forming synapses and growth and development, right? So that's something as children we would certainly need. We've all seen that in disease states, particularly cancer, abnormal overactivity has long been implicated in leukemia. So again, back to the cancer piece, that's been a very informative other branch of medicine, it seems, when we look at neurodegeneration nowadays. Mouse models of Parkinson's disease show promise for neuroprotection looking at tyrosine kinase inhibitors, this type of leukemia drug. However, it's never yet been demonstrated effective in humans with Parkinson's. This is a busy slide. I just wanted to walk you through some big points here. So this talks about sporadic PDA. Again, this is that other group, presumably, if we assume that the GBA and the LRRK2 mutations are responsible for the hereditary forms of Parkinson's. That the sporadic Parkinson's, there is some type of mitochondrial dysfunction. This is the energy-producing part of the cell. There's stress, oxidative stress. We've all heard about oxidative stress for years and years in Parkinson's. It's kind of the generic kind of term for things that damage cells. And what we see that there's increased activated c-Abl protein. And there's a whole lot of bad stuff here that goes on in relation to that, of course. It's trying to handle this oxidative stress. So these are presumably things trying to adapt. Parkin, who's heard of Parkin? Park2. And so one of the hereditary forms of Parkinson's, this actually is part of this pathway of this mitochondrial overreactive kind of pathway here. And there's other inflammatory markers, the microglia, the immune system of the nervous system, the immune cell of the nervous system is activated. And ultimately, cells may die. Progression with Lewy bodies, alpha-synuclein is also implicated. So what is important about this is notice that this is central, at least in this diagram. It's more complex than this, but this is central. So when we're looking for things to slow progression of Parkinson's or get to the root, perhaps cause, or get to one of the root influencers of Parkinson's, we like things that are central, right, down to the core more so. Now we're going to talk briefly about cancer. And this is where, this is so exciting because whenever I see timelines, I always try to think, gosh, where am I on that timeline? Where are my patients on that? Where are we now, right? Because we don't know. We don't know what the future holds. But certainly with the discovery of this Philadelphia chromosome in 1960 began this interest more in the c-Abl pathway as the Philadelphia chromosome was determined to encode this BCR-Abl fusion kinase, 1980. And down the line, they did mouse bone marrow transplantation models of this abnormal protein and developed a drug called Imatinib. Now, geneticists and neurodegenerative disorders, as Dr. Zebatian, I think, have been very influential in discovering genes in neurodegeneration and pathways, but this is a cancer pathway. So thinking about kind of determining cause, determining frequency, determining how relevant a specific therapy might be versus another. And then, lo and behold, we have phase 1 clinical trial of Imatinib presented. Results showing that it influenced the survival in leukemia. And then we start to see the FDA approval, and however, right after approval, what do you see? First cases of resistance reported. Mutations were identified. So the idea of personalized medicine, there really are thousands and millions of types of personalized medicine. And that's the fascinating thing about this. And while even with even if one trial fails, one therapy partially works, we learn a lot from those to go on to the next phase and the next level. And now you have other drugs, Dasatinib and Nilotinib. Who's heard of Nilotinib? Nilotinib, okay. So there is a lot of hype around this medication, Nilotinib. I call it the Nilotinib hype begins. There is a report by Fernando Pagan regarding Nilotinib effects in Parkinson's disease and dementia with Lewy bodies. Well, any first reports on multiple types of disorders is a bit of a red flag, because we're thinking, what is this on? What is it exactly about? You know, and so what was interesting though is this was a non-dopaminergic type therapy targeted at the root cause or a root influencer for Parkinson's disease. And let me walk you quickly through this. This is an open label pilot study to test 2 doses of Nilotinib on safety. This is a fraction of the dose that we use in leukemia, a fraction. We're using 800 milligrams or so. We're talking about 150 to 300 milligrams of Nilotinib. However, this study was really just geared to look at mainly safety, although it was done in advanced Parkinson's patients and those with Lewy body disease, which is a bit of a risky premise, because generally we look at people earlier for safety. But what they did find was, in general, it was tolerated relatively well. The primary outcome for safety and tolerability, it generally met that outcome. There were also what we call exploratory. That means let's see if this helps some other factors of the disease besides just safety in an early trial. And they looked at motor function and cognition. And they did report early on that there were some improvements in both of those areas. 12 patients were in this trial, but they were not compared to a control. And that's very, very important, because what happens is when this trial was released, there was lots of interest in Nilotinib might slow progression to Parkinson's disease. But it wasn't really compared to the control group. It was just compared to where the group started out at. And so we've heard already about, from Dr. Mishley, about placebo and how misleading that can be. So we have to take all of this with a grain of salt. But it was interesting to certainly see that these patients didn't worsen over a 6-month period of time. And also to see that there were some interesting improvements. The mental status score kind of improved compared to the baseline at 6 months. And also there were increased markers for dopamine breakdown at both doses, which is kind of interesting. That's not something that's been measured before. So there were pertinent things to take away from this. But in truth, we really, to this day, know very little about Nilotinib and Parkinson's disease, other than the basic science so far and a bit of the early safety data. Current trials, we have both of these at our site. Nilotinib for Parkinson's disease, so NILO-PD. This is for people with symptomatic Parkinson's disease in one group, and the second group is going to be a longer trial looking at early Parkinson's. And then a brand new trial that's just started from Sun Pharma called Phase 2, and it's SPARK with a C. Our other immunotherapy trial is SPARK with a K. Why this is SPARK with the C, it's a little bit of a mystery given the other trial going on. But this is another TIB medication, presumably, that we think may have a perhaps better safety profile than the first medication. So just in summary, immunotherapy and these tyrosine kinase inhibitors represent 2 new avenues to slow Parkinson's progression. What's exciting about this is we've got a lot of great dopaminergic medicines out there, but we don't have a lot of non-dopamine treatments, and both of these may target more of the critical aspects that cause neurodegeneration. And so far from what's been released, it seems that these may be reasonably safe, but we have to study at the next level, look for a bit more outcomes, look at larger groups of people for more safety, and we hope that Phase 3 trials will be on the way soon. All right, and so maybe we can take some questions. That's all for my presentation. This is just about our group. We've got a booth out there in the Northwest Research.
So I'm curious, if one is positive for a LRRK2 mutation, is that de facto the reason that you have Parkinson's, or is that maybe the reason you have Parkinson's?
So the answer is it's a contributor. It's a big contributor, but as you saw from the slides that I showed, over half of people who have a LRRK2 at least the G2019S mutation and live a normal lifespan won't get it. So it's a big contributor, but it's not the only thing. And the same is true with GBA. GBA is a contributor. It's not as powerful a contributor as LRRK2, but it's a big contributor.
I forgot where I read about the results of the study, but it had to do with the tagging of certain cells for destruction. And they found that the alpha-synuclein molecules were often not tagged for destruction, and they figured out a process by which that could be done, which would eliminate a lot of the alpha-synuclein cells. Have you heard about that? Either one?
Yeah, so one thing I can say is that a lot of, when mutations in different genes are found that cause Parkinson's disease, researchers try to tie that back to synuclein. And so probably synuclein plays a role, it interacts with a lot of these different genes. I think the idea that you just heard about was that synuclein is important for normal function at the synapse, for cells to communicate with each other. The idea is that it's good, but if it aggregates into these clumps, that causes problems with the cell. So anything that can cause synuclein to aggregate, to clump more than it should, is a bad thing. But that could happen by many different ways. With GBA, the idea is that it's causing these lysosomes, this waste removal system, to get clogged up so that synuclein is aggregating and not being cleared as fast, but other proteins are too. So that's one of the ways in which synuclein that's essentially tagged for removal doesn't end up getting removed as fast as it should be.
You know, it's an observation from all of us that are not the professionals, is that some Parkinson patients have a huge battle versus others that have a lighter dose. Is it, explain what you were just sharing with us, all these variances that the individual people have that are in the experiments. It's not that one's got a bigger dose of Parkinson's, it's how their body is susceptible to it.
So I could just speak to one aspect of that. Again, geneticists like me tend to explain everything in terms of genetics. So that's my bias, but let me give you an example. So people with GBA mutations, if you take a group of people with GBA mutations with Parkinson's disease and you take a group of people with Parkinson's disease who don't have GBA mutations, the people on average with GBA mutations get worse faster. What I mean by that is that their motor symptoms get worse faster, their thinking and memory declines more quickly. So it's absolutely true that there are big differences in how fast people decline. Genetics plays a role in that, but there are lots of other things. Dr. Mishley talked about other aspects, how old they are, what they eat, do they take care of themselves, do they exercise, do they have other diseases, do they have high blood pressure, do they have any number of other things. So each person is different, your genetic background, what other diseases you have, what you've been exposed to your whole life that we can't even measure. So those factors probably play a role. It's just that certain factors we can pinpoint and say, aha, you have this, or you don't, but there's lots and lots that we don't understand.
Yeah, I mean, I guess to continue on that point, there are different types of Parkinson's that we recognize, and there's many of those. I mean, you can lump them into people with tremor and those without tremor. And, those without tremor, we see, have a bit more faster progression. It tends to involve cognition earlier, can more aggressively. And I guess the point, I would certainly wonder is that type of Parkinson's mostly affected by like this GBA pathway, not necessarily the gene, but like the GBA pathway. And therefore, even if it's not a gene, but something that influences that pathway, that's going to be the kind of specific personalized therapy for that person. And so that's to your point about like the dose of Parkinson's to get more, it's going to influence or whatever the specific cause of the degeneration or the thing that influences the factors that influence that particular form of degeneration over another type are really where we hope these new therapies are going. And truly, I would caution the group from taking away from this that any one of these might be better than another. It may be that we might find, 20 years from now, look back on the timeline and say, well, there were actually 4 different things we needed to be going after. We were going after 2 at one time. And that's kind of, I think, where we're at now, and wondering, if one of these might, like a GBA, glucose herbicidease GBA therapy, might influence the rapidly progressing patient more than the person with really slow progression. And maybe that works great, it doesn't touch the other patient because there's a whole other different process going on. 2 more questions? We have Mike back there.
Can you hear me?
Yes.
Yes. Genetic testing. So I'll say that actually compared to what we knew 10 or 20 years ago, 9 or 10% is actually a very large number of people. I mean, in the United States, that's probably 100,000 people. That's a lot of people. It isn't everybody, but I think the idea up to this point has been that when people get genetic testing to find out what they have, up to this point, there wasn't really anything that we would do, that information wouldn't change what we would do for you as a patient. It would affect your relatives and whether they were at higher risk for Parkinson's disease or not. But for you, up to this point, it didn't change what we did. It didn't change what trials you were eligible for or what medicines we gave you. Now, that is starting to change. And now the question is, how do we go from where we're at now to being able to screen large numbers of patients with Parkinson's disease in the clinic and elsewhere? And the answer is, we're simply not prepared. There has to be a shift or a paradigm. When you go to your primary care doc, you get a number of standard tests done every time. They check your glucose, they check your white blood cell count, your red blood cell count. So we are going to start to need to do that eventually in the clinic, and I just don't think we're prepared. There's been talk about this, but I think we need to be able to make genetic testing much friendlier, much easier. Another problem is insurance oftentimes doesn't pay for it, and we need to start moving into that direction where we're going to not test you for everything, but at least test you for certain genetic variants that are really important, and do it routinely. You may have been tested, but the point is anybody walking into clinic eventually is going to need to be able to have this option. Okay, so one more question?
Yeah, this is a real simple question. When you talk about early Parkinson's, do you mean early onset or do you mean early age?
So we use the term early in 2 different ways. One is early onset Parkinson's disease, which simply means how old you were when you started having symptoms. And people will sometimes use a cutoff of 50 years or sometimes 40 years. We use the term early in a different context, which is at the beginning of your disease process when you first start showing symptoms, regardless of whether you got the symptoms at age 30 or age 80. So that's, in our parlance, those are the 2 times and 2 instances where we use the term early. One more? One more? I'll tell you what, how are we doing it? We aren't going anywhere, so if you want to come up afterwards, we're happy to talk. I just want to make sure we stay on target. Are we?
Yeah, I'd like to ask about international research. Are you connected with any overseas?
Yes, so we collaborate with researchers in probably a dozen different countries. Some of them are developing countries. Some of those are countries that have well-developed programs of Parkinson's disease. As a geneticist, one of the things that we're interested in is everything you saw here was based on genetics research that was mostly done in people of European origin. We know from other diseases that the mutations in genes that are going to have a bearing on Parkinson's disease in other populations are probably different, and perhaps quite a bit different. So we're interested in looking at African Americans and Latinos. And you all have a brochure, right? Can you turn to the front of it? Anybody recognize who that is? Good. So that's Dr. Ignacio Mata. And he was here at the University of Washington in the VA Puget Sound for over a decade, and he just moved to the Cleveland Clinic. But his career is largely focused on trying to look at the genetics of Parkinson's disease in Latino populations. So that's one of the things we think is really important to do, is to be able to not base everything that you see just on the genetics of people in European-derived populations, because there's other lessons to learn, and we need to get that information in order to be able to treat people from other population groups. It's just lagged behind. So what? No, come up.
Let's thank Thank you so much, doctors, so much. Can you still hear me? I feel like I was louder last time. Maybe my ears are going numb. How's everybody doing? Great lunch? Great. I know, right? Another round of applause for all these amazing presenters. Just a couple short orders of business, and then we're going to have a little break. We are continuing to run a smidge long, which again I see as a positive. It just means we're having lots of good Q&A, and those who you've heard speak, welcome you to come up and ask a few questions if you'd like. They'll also be lingering around after the event so that you can catch them out there. We'd love for you to stop by our sponsor tables, and there's great information and handouts and Q&A that can happen out there as well.